Good afternoon, everyone. I am Dr. Anand Reddy, Assistant Professor in Zoology, Glycolysis B Sc III. Today we will discuss important glycolysis. First of all, objective of glycolysis. First, define glycolysis. Second, which step is involved to the glycolysis process. Third, enzymes are involved to the glycolysis process. And fourth one, function or role of glycolysis. First, definition of glycolysis. Glycolysis means it is the central pathway for glucose catabolism, which is glucose is converted to the pyruvic acid or pyruvate. Another, glucose is converted because glucose is 6 carbon atom and then convert pyruvate molecule according to 10 step catalyzed reactions to form 3 carbon atom of pyruvic acid or pyruvate molecule. Here, first of all, occurrence glycolysis is occur in the cytosol in eukaryotic cell. This takes place also aerobic and anaerobic respiration. It is the process, biochemical process to form energy in the form of energy ATP and NADPH. Here there are different enzymes are included in the process of glycolysis. First of all, hexokinase, second phosphoglucoisomerase, third phosphofructokinase, fourth aldolase, phosphotriose glucose 6-phosphodehydrogenase, phosphoglycerate kinase, phosphoglycerate mutase, enolase and form pyruvate kinase. Here, first of all, glucose is converted to the glucose 6-phosphate, which depends upon ATP molecule. Glucose is added or addition of glucose with the help of ATP molecule to form glucose 6-phosphate, because glucose is 6 carbon atom and these are the phosphate group is attached to the 6 carbon atom to form glucose 6-phosphate and also form to the ADP, adenosine diphosphate, because these are the -1 phosphate can be reduced to the glucose molecule and then form to the glucose 6-phosphate. These are the phosphate group can be added to the 6 carbon atom of glucose molecule, which depends upon hexokinase enzyme. First of all, hexokinase enzymes can be related or helpful for the conversion of glucose to the glucose 6-phosphate. And then another, glucose 6-phosphate is converted fructose 6-phosphate with the help of phosphoglucoisomerase enzyme. These are the isomerase enzymes can be takes place to form glucose 6-phosphate to fructose 6-phosphate. And then another step, third step, fructose 6-phosphate is converted to the fructose 1,6-diphosphate or bisphosphate with the help of ATP, addition of ATP to the fructose 6-phosphate to form fructose 1,6-diphosphate or biphosphate, because one phosphate group can be added to the fructose 6-phosphate. These are the form to the fructose 1,6-bisphosphate. And then these are the 6 carbon atom. And then aldolase enzyme. These are the converted bifurcation to form glyceraldehyde 3-phosphate and dihydroxy acetone phosphate. These are three carbon atom. Three carbon atom glycerol, there 3 phosphate and dihydroxyacetone phosphate, 3 carbon atom. These are the bifurcation can be expressed to the fructose bis phosphatase enzyme that can be formed to the glyceraldehyde 3 phosphate and dihydroxyacetone phosphate. These are the 3 carbon atom. And then glyceraldehyde 3 phosphate is added to the NADH molecule and then formed to the 1,3 bis phosphate phosphoglycerate that can be formed with the help of NADH molecule. Here, role of NADH and role of ATP. There are 2 ADP can be used as well as 2 ATP can be formed during the glycolysis reactions and then formed to the 3 phosphoglycerate, 1,3 bis phosphoglycerate that can be formed to the 3 phosphoglycerate because these are the addition of ADP that can be seen to the phosphoglycerate and then 1 phosphate group can be removed and then formed to the 3 phosphoglycerate. With the help of these are the phosphoglycerate enzyme and then formed to the 3 phosphoglycerate. Another 3 phosphoglycerate that can be formed to the 2 phosphoglycerate because 1 phosphate group can be removed with the help of phosphoglycerate mutase enzyme. Here 3 phosphoglycerate, here 2 phosphoglycerate, 1 phosphate group can be removed and with the help of phosphoglycerate mutase enzyme. And another that can be remove water molecule and depends upon enolase enzyme that can be formed phosphoenol pyruvate. These are the water molecule can be liberated and then formed to the phosphoenol pyruvate. And another step, phosphoenol pyruvate can convert pyruvate molecule. These are the 3 carbon atoms to the form to the pyruvate molecule because ATP can be liberated or removed and addition of the ADP that can be formed to the pyruvate molecule. Here glycolysis can be expressed to the aerobic respiration as well as anaerobic respiration. In the cell, there are formed to the energy which depends upon ATP as well as NADH molecule, and these are helpful for the building block of the cell and as well as building block to the organism that can be seen to the glycolysis. These are the glucose molecule. These are the complex molecule or 6 carbon atom and then formed to the pyruvate molecule. These are the convert 3 carbon atom, the complex to simple. In this process, these are the form to the 3 carbon atom. Here, reaction can be expressed during the glycolysis process. C6H12O6, these are the glucose molecule. Carbon atom 6, hydrogen atom 12, oxygen atom 6. Here, glucose 6 carbon atom to form to the 2 pyruvate molecule. 2 pyruvate molecule. Here 2 ADP can be used, 2 phosphate group can be used, 2 NADH molecule can be used during the process of glycolysis and then form 3 carbon atom, hydrogen 4 and O oxygen 3 atom that can be formed to the after end of the glycolysis. Here 2 water molecule can be liberated, 2 ATP molecule can be liberated, 2 NADH molecule can be liberated and 2 hydrogen atom can be liberated. These are the 2 ATP can be formed and 2 ADP can be used during the process of glycolysis. According to the glycolysis process, they form to the pyruvate molecule. Means these are the glucose is converted to the pyruvate molecule. According to these are the 10 enzyme catalyzed reactions. Enzymes can be used: hexokinase, phosphoglucoisomerase, phosphofructoisomerase kinase, aldolase, phosphotriose isomerase, glycerol 3-phosphodehydrogenase enolase, aldolase, phosphoglycerate mutase, enolase आणि pyruvate molecule. These are the all 10 enzymes can be used to the process of glycolysis. Here pyruvate molecule can be 3 carbon atom that can be formed to the glycolysis. During anaerobic process, these are the form also in the fermentation process, and in aerobic process, these are the form to the pyruvate molecule. These are the fermentation process that can be lactic acid will be formed to the convert according to the pyruvate molecule. And then aerobic respiration, these are the glucose is converted to the pyruvate molecule. It depends upon enzyme catalytic reactions. And role of glycolysis. Glycolysis means glyco means glucose. Glyco means glucose. And lysis, these are the debris or these are the reactions, 10 enzyme catalytic reactions or breakdown to the glucose molecule. Here function or role of glycolysis, these are the helpful for the cell build up to provide energy to the cell in the form of ATP, NADPH molecule, and these are the helpful for the all cell or eukaryotic cell during the aerobic respiration. These are all glycolysis can be useful for the aerobic respiration because these are the form to the in the form of ATP, adenosine triphosphate, to the formation of energy to build up cell. And here, according to the glycolysis enzyme, these are that takes place to the conversion of one step to the other step. Glucose is converted to glucose 6-phosphate and fructose 6-phosphate. And these are the different enzymes. 10 enzyme catalytic reactions are seen to the glycolysis process. Here, catabolism. These are the two metabolism can be takes place in our body: anabolism and catabolism. Anabolism and catabolism. Here, catabolism is, suppose for example glucose is 6 carbon atom. These are the complex food material is converted into simple food material. These are called as a catabolism. Means glucose is converted into pyruvate molecule. These are the 6 carbon atom liberate to the 3 carbon atom. These are the form to the catabolism. And anabolism means these are the simple food material is converted to the complex food material. These are the called as a anabolism. These are the difference between catabolism and anabolism. Here glucose is catabolism means 6 carbon atom. These are the complex, and these are the convert to the pyruvate molecule to the with the help of different types of enzymes. 10 catalytic enzymes are seen to the glycolysis. These are the important role to the all aerobic and anaerobic respiration, and these are the helpful for the curriculum in BSc Part 3rd to discuss very important topic in glycolysis, and these are the role and application as well as steps of enzymes, different types of enzymes, occurrence, and as well as definition of this glycolysis. These are the very important, and then chemical structure that can be seen also. These are the benzene ring structure, 6 carbon atom, and then form to the 2 carbon atom pyruvate molecule, and these are the 6 carbon atom. How can phosphate group add, remove, addition and deletion of phosphate group, and then form to the ATP, and which how many ATP can be used, how many ATP can be formed, as well as how many ADP can be used, how many ADP can be formed during the process of glycolysis. Here 2 ADP can be used and 2 ATP will be formed during the process of glycolysis. 2 phosphate group can be used because addition of phosphate group, these are the 1,3 bisphosphate or diphosphate, glucose 6-phosphate, and 2 NAD molecule. nicotine adenosine diphosphate, and then 2 auto molecule can be liberated and 2 ATP will be formed and 2 NADH molecule will be formed and 2 hydrogen molecule can be liberated during the process of glycolysis. Student can be seen or asked the questions in the university exam, how many ATP can be used or how many ATP can be formed during the process of glycolysis. NADP molecule can be used, NADPH molecule can be formed. But these are the important role to the glycolysis.